Hello and welcome everybody to my channel Omini Bio Entrance. Today we will be discussing the National Entrance Screening Test NEST 2020 session 2. I have discussed only the biology. Coming to the first question, a selenium activated protein P catalyzes a metabolic reaction that results in release of two H positive ions. Expression of P is induced by lactose. In an experiment, cells were grown for 1 hour in selenium and lactose containing media, resulting in a net concentration of 10 raised to 6 molar H positive per cell. Selenium and lactose were removed from the media and cells were grown for another 10 generations. Assuming that all other proteins are expressed at constant level, the net change in pH inside the cell at the end will be, answer is 3. Next one, a drug P inhibits DNA replication in the bacterium E. coli by specifically blocking the catalytic site of DNA polymerase activity. That the same drug P can be used to treat cancers in humans can be explained from the fact that because mechanism of DNA polymerase is majorly conserved. That is why, because the drug is blocking the catalytic site of DNA polymerase activity. So the mechanism of DNA polymerase is majorly conserved in all from bacteria to humans. Then P can be used as a drug to treat cancers in humans. Then endoplasmic reticulum is tubular, Golgi apparatus is stacked, flat cisternae, mitochondria are oval shaped, however lysomes are spherical in shape. This fundamentally result of the propensity to minimize surface tension. Then next one, proteins in biological system are polypeptides that are exclusively made from L amino acids. A peptide P with L amino acid of a particular sequence always adopts a right-handed alpha helical structure. Under identical condition, if all the amino acids of the peptide P are changed to D amino acid, then the expected conformation from the options given below would be, that is option 4. Why? Because D amino acids form left-handed helix. Then next one, a polymerase chain reaction for amplification of DNA fragments involves multiple cycles of thermal denaturation, annealing and extension steps in each cycle. Because of the heat denaturation step, a thermostable polymerase is required for performing PCR. And thermostable polymerase, that is Taq polymerase, is isolated from Thermus aquaticus. An experimenter had all the resources except the thermostable DNA polymerase to perform a PCR. The experimenter can use dash to substitute for thermostable DNA polymerase. He can use an Escherichia coli DNA polymerase added after the annealing step of every PCR cycle. Why you have to add it after annealing step of PCR? Because the first denaturation temperature is very high and E. coli DNA polymerase is not thermostable. Hence, it has to be added after annealing step of every PCR. Then next one. Conjugation in E. coli is a microbial process wherein conjugative plasmids are transferred from one bacterium to another that does not possess it. This requires physical contact between the cells involved in the transfer. In an experiment performed in a sterilized U-tube, strain P, resistant to ampicillin encoded in its plasmid, and Q, resistant to tetracycline encoded in its genome, were grown in an appropriate medium without an antibiotic. A 0.22 micron pore size nitrocellulose membrane placed in between the two arms serve as a partition. In a parallel identical experiment, the 0.22 micron membrane was replaced by a 30 micromolar nitrocellulose membrane. After allowing enough time for the conjugation process to occur, an aliquot from each arm is plated on either ampicillin or tetracycline antibiotic containing the plate. Choose the correct statement about the outcome of the experiment. So they have taken a U-tube. On one leg it is strain P and on the other it is strain Q. Similarly, for experiment 2, they have taken strain P and strain Q. The difference is that in experiment 1, 0.22 micron filter is used, whereas in experiment 2, 30 micron molar filter is used. So 30 micromolar is having more pore size than 0.22 micron filter. So in case of experiment 1, there will not be any physical contact for the transfer of plasmid between strain P and strain Q, whereas here in experiment 2, there will be a physical contact between the strain P and Q and conjugation will take place. So first one, strain Q would be ampicillin resistant in experiment 1. No. Why? Because the 0.22 micromolar filter doesn't allow physical contact between the two bacteria. Then strain P would be tetracycline resistant in experiment 2. Conjugation is for transfer. They have told in the question that it is meant for transfer of the plasmids, not for genomic DNA. So tetracycline resistant gene is present in the genomic DNA. So strain P can become tetracycline resistant in experiment 2. Then strain Q would be ampicillin resistant in experiment 2. Yes, because there is physical contact between the two strains, because the pore size of the membrane is 30 micromolar and P will transfer its plasmid for ampicillin resistant gene to strain Q, and strain Q would become ampicillin resistant in experiment 2. So that is correct. Strain P would be tetracycline resistant in experiment 1. No, that is also. Then next one. Graphical representation of action potential across the membrane of a neuron is shown below. The different stages of the action potential are shown with the numbers 1 to 5. Choose the correct statement about the Na plus K positive ion concentration across the membrane in stage 4. So membrane, this stage 4, it represents membrane repolarization. So during membrane repolarization, it results into formation of for rapid sodium channel inactivation, as well as a large efflux of potassium ion resulting from activated potassium channels. So option 1 is correct. Opening of K positive ion channels leading to K plus efflux. Then next one. Height of a plant is positively determined by the amount of product K of a metabolic pathway catalyzed by 5 enzymes L, N, M, P, O as depicted below. The intermediate product I blocks the activity of enzyme P, whereas intermediate J blocks the enzyme L. Conversion of I to J is the rate limiting step. Based on the information, choose the correct statement from below. Silencing the gene M will result in tall phenotype. No. K will not be produced on silencing the M. Then silencing of the N gene will result in tall phenotype. No, it will not produce H, I, J, K products. Will not be produced. So that is also wrong. Overexpression of P gene have no effect on height of the plant. Once P gene is overexpressed, it will result in the production of more I, which in turn will block the activity of enzyme P. So more I will also result in the production of J, which will also inhibit the L enzyme. Since conversion of I to J is the rate limiting step or the slowest step of the pathway, and overexpression of P gene will not have any effect on the height of the plant. So option 3 is correct. Next one. On an oceanic island, long isolated away from a continent, there are only 4 coexisting species: flowering shrub, rabbit, large deer and small fox. The food web for the island ecosystem is depicted below. Deer and rabbit eat precisely the same part of the flowering shrub, depleting the shrub vegetation at precisely the same rate. If predation is not the reason that the 4 species coexist, choose the statement that best explains their coexistence. So option is intraspecific competition is stronger than interspecific competition. What is intraspecific competition? It is an interaction in population ecology whereby members of the same species compete for limited resources. This leads to reduction in fitness of both individuals, but the more fit individual survives and is able to reproduce. In the question, flowering shrub is eaten by both large deer and rabbit, and this can cause a competition between the deer and the rabbit community for the same source of food, as well as there will be competition within the rabbit and deers among themselves for the same resources. But this intraspecific competition is more than interspecific competition, and it will be stronger. So members of the same species have rather similar requirements for resources, whereas different species have smaller contested resource overlap, resulting in intraspecific competition being more stronger than interspecific competition. So option 4 is correct. Then next one. Choose the correct statement from the following regarding light reactions taking place in chloroplast. The H positive ion concentration of thylakoid lumen is higher than stroma. That's correct. Flow of H positive ion through ATP synthase drives phosphorylation of adenosine diphosphate. That is also correct. The electrons for photolysis reaction of water are passed to the photosystem too. So 1, 2 and 3 all the statements are correct. So option 4 is the answer. Then next, a crop variety was developed at a research station in Dehradun, located a temperate zone, and this variety produced round or wrinkled seeds with green or yellow seed color. A field test was performed with the seeds from a cross between round and green and wrinkled and yellow parents. Seeds obtained from this test were planted at the same research station and compared to 3 new tropical locations with different levels of microbial infection and salinity changes. Based on the information provided and assuming that the genes follow Mendelian inheritance with no other factors affecting the yield, choose the correct statement from the option given below. I have given it in the next slide. Now what they have done was they have produced seeds from F1. So the total yield represent the F2 generation of seeds. So whatever is given here, that is the number of seeds that germinated, that will belong to the number of seeds sown will belong to the F1 generation, and when you get seeds or plants that produce seeds from the F1 generation, that becomes the F2 generation. Okay. Now, option 1. It is better to sow 3 parts of green seeds and 4 parts of yellow seeds in tropical region. That is wrong. Then wrinkled character is better suited for tropical region than round character. That is correct. Then only yellow seeds are better to sow in the tropical region carrying high microbial infection load. In temperate regions, the green plants were more than yellow, whereas in the tropical region, at whatever growth condition, yellow was more than that of the green color. So even when there was a high microbial load, yellow seeds survived and adapted to it more strongly than the green ones. So option 4 is correct. So 2 and 4 are the answers. Then next one. In order to understand immune responses to COVID-19 viral infection, a researcher took 5 individuals of an animal model of the disease. One of these animals was infected in controlled lab conditions by nasal spray of the viral suspension. The animal developed pneumonia-like symptoms, fever and difficulty in breathing. 10 days later, blood plasma was collected at 2 time points from the infected animal. When the symptoms were still there, draw 1, and 2, 7 days after the animal completely recovered from the illness, that is draw 2. The remaining 4 animals were given different treatments with the plasma, either draw 1 or draw 2, and then challenged with viral infection. Symptoms development was monitored and the results are tabulated. Assume the plasma is made hypoallergenic by chemical treatment. Now based on these results, the correct statements would be: in treatment O, the viral antigen from the plasma generated a humoral immune response in the animal and thus protected it. It is not humoral response, it is just passive immunity. So option 1 is wrong. In treatment M, fever was because of cytokines such as interferons that are in the plasma. That is correct. That is why the injection of draw 1 followed by mock spray, that is no viral particles were there. Only fever developed, that was because of cytokinins such as interferons in the plasma. Then in treatment O, antibodies COVID-19 gave passive immunity to the animal. I already told when we read the first option. Then in treatment N, symptoms were mild because interferons from the plasma set a general innate immune state in animal. That is also correct. So option 2, 3, 4 are correct. Now next one. Hydra has a receptor Q in certain cells that respond to a specific neurotransmitter. P1 isolated from brains of higher mammals. An experimenter finds that many species have neurotransmitters that are structurally related to P1 and have functional homologs of receptor Q. Binding of P to Q elicit neuronal response. A summary of this study is given below. Based on this information, choose the statement that is likely to be correct. In mammals, P3 can compete with P1, P2, P4 for eliciting the response. The question does not mention regarding interaction of P1, P2, P3 and P4 with each other. Then, insects might have lost the gene Q due to loss of endogenous production of P group of neurotransmitters. That can be true. Insect might have lost gene Q because they don't have any P1, P2, P3, 4 molecules in them. Then, insects have lost the gene Q after fishes have evolved. Yes, that is correct. Otherwise, fishes would not have had the gene expressed in them. Insects evolved into fishes. So only after the fishes evolved, insect lost the endogenous production of P group of neurotransmitters, resulting in loss of the Q genes. Then, next one. In mammals, the Q gene multiplication occurred as a result of diversification of the neurotransmitter P. Yes, that possibility cannot be eliminated, and it is true also because there is the presence of P group of molecules in other animals lower to mammals. So other animals are having P1, P3, P4 in them. So when mammals evolved from other lower animals, they also carried the P1, P2, P4 response gene. So the Q gene multiplication occurred. That is, the number of Q genes increased in case of mammals as a result of diversification of the neurotransmitter P. So option 4 is also correct. 2, 3, 4 are correct. Then next one. Antibiotics P1, P2, cell wall synthesis inhibitors and Q1, Q2, protein synthesis inhibitors are equally hydrophilic and moderately toxic to humans. Staphylococcus is known to develop resistance to all these antibiotics. An experimenter tested these antibiotics against a recent pathogenic strain of Bacillus. A high density of Bacillus culture was spread on nutrient agar plate. Filter paper discs, each soaked in P1, P2, Q1, Q2 individually of the same concentration, were placed on the bacterial lawn. M is mock control without any antibiotic. Following an overnight incubation, a zone of inhibition is seen around each disc except for M. Additionally, few individual colonies were observed in zone of inhibition for P2. Based on these observations, select the correct statements. Now what they have done is there are two antibiotics, that is P1, P2 and Q1, Q2. Now these were checked for the pathogenic strain of Bacillus species. So what they did was a high density of Bacillus culture was spread on a nutrient agar plate. Then these filter discs which were soaked in P1, P2, Q1, Q2 were placed on top of this bacterial lawn. Now M was taken as the control, that is it didn't have any antibiotic. So naturally it will not give any zone of inhibition. The next day when they took out the Petri plate, they found that P1 is having a zone of inhibition, P2 is having zone of inhibition, but some colonies are growing in P2 zone of inhibition. Then Q1 also is having a zone of inhibition and Q2 is also having a zone of inhibition. The diameter of the zone of inhibition in all the four is different from one to the other. Then based on these observations, select the correct statement. Q2 would be the preferred antibiotic for therapy against this bacillus infection. That is true because the zone of inhibition of Q2 is having more diameter than the others, that is P1, P2 and Q1. And moreover, P2 zone contains resistant colonies growing in the zone of inhibition. So that is not a good antibiotic. So Q2 is better or preferred antibiotic for the therapy. Then if P2 is a new generation antibiotic derived from P1, then long-term use of P2 is likely to remain highly effective against the bacillus infection. This statement is true because P2 zone contains resistant colonies growing in the zone of inhibition around P2. Then the combination of P1 and P2 will not lead to antibiotic resistance in the bacillus. P2 will lead to antibiotic resistance in bacillus. So that is also wrong. Then the combination of P1 and P2 is least likely to lead to antibiotic resistance in the bacillus. Yes, because the diameter of the zone of inhibition is Q2 is more, and also no resistant colonies are growing as in P2. So the combination of P1 and P2 is likely to lead to antibiotic resistance in bacillus. Sorry, it is least likely to lead to antibiotic resistance in bacillus. Then next one. Barbara McClintock observed non-Mendelian pigmentation pattern in maize mediated by transposons. Transposons are jumping genes, as also observed in petunia. In petunia also you can see different colorations in the petal because of jumping genes. These somatic changes were inheritable as evident from the clonal pattern of pigmentation observed both in seeds and flowers. So these are not heritable changes, are inheritable. As leaves grow, jumping genes get activated and insert themselves into chlorophyll producing genes, thereby disrupting and inactivating them. So jumping genes will get inserted into chlorophyll producing genes and they will disrupt the formation of the green coloration in plants, which is given by chlorophyll. This is typically seen as leaf pigment variegation, and this you can see as variegated leaves. That is, some dots of green will be there and also white coloration will be seen on the leaf, as you can see in P and Q. Sometimes the transposon jumps out from where it was inserted, restoring the chlorophyll gene function. So the transposon will also jump out from where it was inserted in the chlorophyll producing gene, thereby restoring the chlorophyll gene function. Observing the leaf variegation patterns in P and Q, choose the correct statements. Only Q shows clonal propagation of the variegated phenotype. Now what is clonal propagation is nothing but multiplication of genetically identical copies of a cultivar by asexual reproduction is called clonal propagation, and a plant population derived from a single individual by asexual reproduction constitutes a clone. Option 1 doesn't mention anything about clonal propagation in the question. So option 1 is wrong. Then only P largely indicates that the transposon can jump out of chlorophyll gene resulting in function. Yes, you can see some spots with chlorophyll and some without chlorophyll, showing the jumping character of the gene. Then tissue culture propagated plants from greenhouse tissue of leaf Q will show no variegation. The question does not mention about tissue culture. So 3 is also wrong. Then both P and Q show sporadic events of transposon-mediated variegation. So what is sporadic? Sporadic means occurring at irregular intervals or only in a few places, scattered or isolated. So P and Q show this character. So option 4 is also corrected. So 2 and 4 are correct.